Hi everyone, welcome to chapter 3. This chapter is all on biology and neuroscience, and we're going to do a few different things here. We're going to talk about neurons and how neurons work. We're going to talk a little bit about different parts of the nervous system. We'll also get into a little bit about parts of the brain and how those are sort of related and what some of those do. And also we'll talk a little bit about how we study this from a neuroscience perspective and some tools and things that we use to do that. All right, so let's go ahead and jump into this here. So again, chapter 3, biology and neuroscience. We're going to start off this section talking about neurons, or how neurons kind of work and what they do. So neurons are structured in kind of a cool way. And we can see here some of the unique things about them and some important things to kind of understand how they work. But generally what neurons are is the communication devices within our bodies. And so they send signals back and forth, and we use these to kind of communicate information from one place to another. And so some of the most important things I want to kind of point out about neurons is, again, first off, their physical structure. And so as you can see here, this is an example of one type of neuron where we can see we have sort of that cell body located sort of in the center area here. And from that cell body, we have these things kind of coming out, protruding from that cell body. These are called the dendrites. And these are responsible for receiving signals or receiving information from other areas or other neurons that they're communicating with. So the signal comes in, again, through these dendrites from other neurons that is sending information to this neuron. And then that signal travels down what's called the axon. So it travels down the axon and ends up at these parts of the neuron, which are called the axon terminals. And at the very end of those are the axon buttons. And those are what actually send the signal or pass that information along to the next neuron in the chain of communication. And so we sort of see this general overview of how this information travels through the neuron and down to the next neuron that's connected to it. And so if we think about this sort of in this, sort of linear fashion, we could see these axons connect to, say the dendrites of other neurons, which have their cell body and send that down to the next set of neurons. And so this change just kind of happens, and there's connections to all different types of neurons that happen. So, you might be receiving information from one neuron here and a different neuron here, and these networks of information connect to be able to pass along complex pieces of information and overall, communication between different parts of the body. And so another piece of this I want to point out is this axon area here, right, where this long chain is, where the information sends through the cell. We see some things around it. So we see sort of these like bubble things that are appearing here. These are called myelin or myelin sheaths. And what these do is they act to kind of speed up that communication or speed up that signal that gets passed through the axon. And so one way to think about this is, if you think about this without those myelin sheaths, the signal would have to pass all the way along this entire length of the axon. And so that can be long distances, or that is just a longer distance. Then what it can do using these myelin sheaths is it can actually kind of jump between these and take a shorter path. So that signal, that electrical signal, kind of jumps between those myelin sheaths and can send that signal faster and more efficiently down the line. And so some things that we think about when we think and understand kind of what this does is, this is sort of a fatty tissue. And so there are things that benefit this fatty tissue growth and development, certain foods, certain, healthy lifestyle and exercise benefits this growth and this support system for this myelin sheath. And we see this as evidence when we see, healthy cognition and things like that. And so as we age, as we get older, we see that myelin sheath start to break down. Certain cognitive degenerative diseases, we see that breakdown of the myelin sheath, which slows down that signal process and that cognitive sort of, communication it just slows down without that myelin sheath. And that's one of the reasons we see this happen in older age and in unhealthier lifestyles. So that's generally the overview of the neuron. Let's talk now about how neurons communicate. So how that happens between neurons. So within the neuron itself, talking about that signal that we just mentioned, that's an electrical signal. Between neurons, so where they connect, we see sort of this example here where we have the neuron button or the terminal button of one neuron and the dendrite or the receiving side of another neuron. And so what happens is in between these communication points, we actually don't have an electrical signal being sent, but a chemical signal that's being sent. And so inside this gap here that we see, there's actually some space between the two neurons communicating with one another. And in that space, they use what are called neurotransmitters or chemicals to pass along and send that information. And so we can see some examples of what that looks like here, where those neurotransmitters get released from the terminal button and get absorbed or received by the dendrite. And that's how that information passes along from one to the next. So it's a really cool thing. And we're going to talk a little bit more about different types of neurotransmitters, but we'll see that neurotransmitters act in a lot of different ways. They could be excitatory, they could be inhibitory, whether they're trying to get the next nerve or the next neuron to fire, or they're trying to keep it from firing. They work in a lot of different ways to kind of see that communication network as it kind of gets built. But lots of different types of neurotransmitters as well, and they exist in different regions of our brain and our body to kind of do different types of things. So we'll talk a little bit about those as we go too. So next, let's talk about sort of how the signal actually gets sent and how it actually fires within the neuron. So an important distinction to make here too is that inside the neuron, like I said before, this is an electrical signal that's being sent. And we talk about what's called the all or nothing principle, which means that the signal or the neuron is either going to fire or it's not. So there's no variability, there's no amount of it firing a lot or firing a little bit. It either fires to its full strength or it doesn't fire at all. And so we can see that kind of here in this graph, where we can see as our resting state becomes higher and higher and higher, eventually we get past some threshold mark. And at that point, we get what's called depolarization, and we get that firing of the neuron. And it's always going to fire to that same height, no matter what. And then it comes back down, repolarization, to some refractory period where it can't fire again for a period of time until it again gets back to its resting state and can start this process over. But an important distinction here, like I said, is that this peak, this height, the strength of the firing is always going to be the same no matter what. It doesn't vary. So it's not like it's sending a strong signal or a weak signal. It's just sending its one unique signal. And so why this is important is it brings up the question of, well, how does different information gets passed along? How does that information maybe change or vary in types of signals? And so what actually happens, which is really cool, is rather than the strength of the signal changing, the speed of how frequent the signal gets sent is what changes. So maybe a neuron can fire really rapidly, really fast, and that tells some piece of information differently than if the neuron is firing slowly and only sending out a signal every once in a while. And so this is how information kind of is modulated and is altered, is not by the strength, but by the speed of how fast it fires. Now, I don't want to spend too much time on sort of the sodium ion channels and how this actually happens with the polarization. The main thing I want to just kind of address is very simply that these do exist, and how that polarization, how that balance in charge within the cell changes is through sort of these membranes and through these sodium ion pumps. And so generally there are chemicals that hold different levels of charge, and the cell is going to be sending some in and some out of it over time, and that is going to trigger it to either polarize and have this positive charge, which is what sends the signal. And so just know that this is sort of how this works, but we're not going to get too far into the details of this beyond that level. So I also want to talk a little bit about how drugs sort of mimic different types of neurotransmitters, and also just a little bit about how neurotransmitters themselves work. So I said before that neurotransmitters are sort of what we use to communicate from one cell to another or one neuron to another, and that is sort of the chemical process of sending information. And so we can see here that it really does act sort of like this lock and key. And so when we have those neurotransmitters, they have some particular shape, some chemical property that produces some shape of how they exist. So again, let's say we have sort of our terminal button up here with our neurotransmitters that are being sent out. And let's say these are our neurotransmitters coming out into that synaptic cleft, or that in-between space between the terminal button and the receptor, or the dendrite of the next cell. So we'll just say this is sort of the dendrite. And the dendrite has sort of these receptor sites on it to receive certain types of neurotransmitters. And as these float and are released, if they have the right shape, the right key, they fit into the correct lock and they pass on that signal. If, on the other hand, they're the wrong type of neurotransmitter, well, they're not going to fit into that receptor site and they're not going to pass on that signal. And so having the right type of neurotransmitter and having the right type of receptor site is important for that signal to pass along in certain regions. And so what's really interesting is what drugs and different chemicals can do is they can act in a lot of different ways. Either they could come in and they could mimic the shape of, say, a neurotransmitter and add more of it into this equation, so it's binding more and more to those receptor sites. Maybe it could actually act in its own shape where, maybe it has some unique property to it that can block those receptor sites but doesn't actually send the same type of signal. So it can mimic, some pairing, but also block the ability for the real neurotransmitters to bind, which changes, again, that type of signal being sent. So again, I don't want to get too far into this. There are lots of different types of drugs that act in different ways, but I find it really interesting to sort of understand how neurotransmitters work in this way and see how we can kind of mimic and alter and change this process through these different types of chemical agents that we can sort of alter.